Alright, still in Chapter 3, we're talking about antibodies and antibody fragments, and the fact that antibodies can serve as antigens. So you can inject someone with antibodies from a different animal or a different human and actually raise anti-antibodies against them. Okay, don't get too confused. You can raise antibodies from a mouse to a goat, or from a human to a goat. You can get anti-human antibodies from injecting human antibodies into a goat. Think about it for a minute. I think it'll make a lot of sense. There are different classes and subclasses of antibodies that are there. There are three main monomers of antibodies: IgG, IgD, and IgE. I usually just remember that a GED is kind of the first step of education. So, one, these are individual subunits. Then an IgA, if you write the capital letter A, it's got two straight lines as legs, so it's a dimer. And then IgM, and if you look at an M, it's got 5 points on it. Those 5 points lets me know that it's a pentamer. And as you read through this, you'll see what some of the functions of them are. But the thing that's going to trick you, that you're going to have to really pay attention to, are the fact that you can have antibodies raised against them. So the anti-immunoglobulin antibodies have been extremely useful in studying antibody structures and setting up things like the ELISA test. So you're going to have to try to differentiate and learn. And I could sit here and tell it, but you can read it just as well. What is an isotype? What is an allotype? What are idiotypes? So there are different types of antibodies that can be produced against another animal's antibodies. So that's extremely useful in research. Read carefully the different types of antibodies, when they're produced, where they're produced. For example, IgM and G are some of the most common ones you find in blood serum, where IgA tends to be a secretory antibody, so you find it secreted into mucus, in your lungs, lining your digestive tract, earwax, places like that. So read about the antibodies that are there, and I think it ends on page 90, and then it's going to talk about X-ray crystallography as the next thing for antigen-antibody binding. You can look at some of those figures, and it's kind of interesting to look at the binding. It's not just three-dimensional shape. It's the electrical charges that are given between those organisms at specific pH regions. So you can have antibodies that can function very well in the kidneys or in the lungs, but they wouldn't function in other areas of the body because of the different pH ranges that are there. Alright, so the rest of the chapter is going to be talking about B cells and their signaling, and then T cell receptors and signaling. Pay very close attention to the generation of antibodies specific to TCR on page 96. That figure is a really good figure to look at, and I may use something similar to that on the exam. I'm on page 98. I'm not going to talk too much about the signal transduction complexes. Just know that they've got to come into contact with one another, and that's where you get the lipid rafts and other things that can influence how often these things come together. Now on page 99 is where we pick back up and we start having to pay closer attention. So look at figure 3-31. Read the section that says the T cell co-receptors CD4 and CD8 also bind the MHC, because that interaction is important for the rest of the semester. I'm not going to make you learn the lick and first tyrosine kinase activated T cell stuff. Just know that the signal transduction has happened. ZAP 70, none of that is really that important. If you need to know that stuff, you can look it up. But you do need to understand general structure of the CD4 versus the CD8s, and that these are co-receptors. It says core receptors for some reason. They're immunoglobulin-like domains, and there's a lot of similarities between these and other molecules of their type. Okay, well, that's it for Chapter 3. Next, we go to Chapter 4.